revision stream. Well, that's a lie. No, not the last one. One of the last ones we'll be doing before paper one, which obviously is happening on Monday. Oh man, it's weird. It's weird because in some ways I really am looking forward to this. I'm looking forward to, getting it over and done, but at the same time, man, if certain topics come up, I'm gonna be crying, man. I'm gonna be crying on that paper. So what we're doing today, just as a quick summary, is I'm going through one of the 2020 papers. I'm not going to be answering many questions. Instead, we're going to kind of be making a couple notes here because we have access to the examiner reports. And basically what they are is they are the examiners explaining how people have messed up the question. And a big thing that I do is I mess up questions. I will overthink them. I will write about the wrong thing. I will do something wrong. I do it all the time. We all do. So let's take a look at this paper and let's see how we can fix up. So the first question here shows that is alpha glucose, a amino acid, adenosine triphosphate, and beta glucose. And what it's basically telling you here is match it up to the statement. So this is a very nice format question where you literally just have to match up. So is a monomer in an enzyme's active site? Enzymes are obviously proteins, so that is going to definitely be the amino acid. Is a monomer in cellulose? That's obviously beta glucose. Forms a polymer that gives a positive result with a Biuret test, purple protein. So that is amino acid, and is produced during photosynthesis and respiration, ATP. Nice and easy question. Let's see what the examiners said. It may be worth noting that for question 1, students could use a letter once, more than once, or not at all. Okay, so right there, they've just admitted to baiting people because, I mean, come on, you look at that and you're likely to think, hold on, I missed out on alpha glucose. Which one's alpha glucose? None. It's a bait question. Let's go to question 1.2, see what they said. Oh, this is the raffinose question. This is the question where you get given the formula of 6 carbon, 12 hydrogen, and 6 oxygen. And what it's telling you is that there is a polymer which is basically this repeated 3 times. And it's asking you, what would the formula be? So if each one of these little, hexagons, if each one of those has this formula, what would the overall formula be? This is such a big question. I love it. Now, again, I messed up this question first time I did it. I completely messed it up. Because you guys need to understand something. Condensation. Condensation is the joining of chemical bonds by eliminating a water molecule. Water. So actually, the formula would be, if we lived in an ideal world, C18, H36, or 18. That's like the ideal world right there. Everyone probably put that. But what you're forgetting is that it's bonded. There's glycosidic bonds. There are 2 glycosidic bonds. So that's 2 water molecules. So you would remove, and you would remove. And that's how you get your answer. Just going to check if that's right. Yep, C18, H32, O16. So let's see what the mark scheme said. It was surprising how few students could gain the 1 mark. Okay. So a lot of people got that wrong. Likely because they did the mistake that I did and just multiplied it by 3. No, it's 3 molecules, but 2 condensation bonds. That's the... So yeah, honestly, I would argue this is a really scummy question. I got this wrong the first time I tried it. In an exam, I was able to get it right, but only after I realised what happened. Only after I realised how you did it. 3 marks. A biochemical test for reducing sugar produces a negative result. Describe a biochemical test to show that raffinose contains non-reducing sugars. Alright, this is easy. This is nice and easy. For 3 marks, I would love this. That's 8 marks so far. Peak. I mean, in an exam, ideally, I would get more, right? But who knows? Maybe I made a mistake on that question. But anyways, mark point number 1 is hydrolyze. Or basically, not hydrolyze, sorry. That is what happens, but to do that, you would add acid, and neutralize. Honestly, okay, hold on. I say this is easy. In a water bath. And a positive result. Add Benedict's solution. A red precipitate indicates a positive result. I just want to make sure I got the wording right there. Heat with acid and neutralize. Heat with Benedict's solution. Red precipitate. Okay. So, heat with acid and neutralize in a water bath. Add Benedict's solution. That's your key point. It would allow you to say accept boil/water bath, but you would have to specify that you do it for both. So I might have only gotten 2 marks there because I didn't specify. I mean, if I started it by saying in a water bath, add acid and neutralize, then add Benedict, I think I would have gotten all 3 marks. So I might have dropped 1 mark there if I had a really bitchy examiner that, stuck by the mark scheme like it was a code of honor. Let's see what they said. Let's see what the examiner said. Many students were able to score at least 1 mark, although very few could clearly and correctly write down the practical steps. See, I wouldn't have thought that, but then again, maybe people wrote just about the Benedict's test. Remember, guys, if you're absolutely in doubt about the tests, if you're absolutely in doubt, you have no idea what the steps are, at least talk about a positive result. You will always get a mark for talking about a positive result. So in the emulsion test, that would be I think you have to say milky, you can't say cloudy. A milky white emulsion. That's what happens in an emulsion test if it contains lipids. In a regular Benedict, it will form a red/orange precipitate. And in a Biuret test, it will form a purple precipitate. This is common knowledge, and just very quickly, the steps for the emulsion is you add ethanol, add water, shake, and then you have to talk about the positive results. Benedict, you either just add Benedict, or I should say in water bath. In water bath, add Benedict's. If it's a non-reducing, you will also have to add acid and neutralize before then adding your Benedict solution. Because the idea here, if you're asked to explain why would you add acid, you would need to say that hydrolyzes the glycosidic bonds in the non-reducing sugar. And neutralizing it, obviously, removes the acidic conditions so that the Benedict solution can work. In the Biuret test, you literally just add Biuret. So there you go, a quick recap of the food tests there. I know there is another test for lipids called sedan-free. I think you need to do that in a water bath, I'm not sure. But for that one, you would expect to see a red line at the top. But anyways, that's not what we're doing, so whatever. Explain the arrangement of phospholipids in a cell surface membrane. For 2 marks. Alright. I'll be honest, I might get this one wrong. Let's see. Let's firstly draw the phospholipid. Okay, so what we have here is you have the one kinky chain, that's the unsaturated, that's the saturated fatty acids. You have your, this would be the glycerol, and your phosphate group. So obviously, this is going to be the polar head, that's the non-polar. And the idea you need to talk about there is this is hydrophobic. This is hydrophilic. So I guess firstly I'll say hydrophilic, polar, should I say head or I'll just say glycerol. Polar glycerol head faces, I'm gonna say fluid / water. Because I don't know specifically what it would want you to say there, so I'll underline that. Hydrophobic, non-polar fatty acid chain, face inwards away from water. And it says the arrangement of phospholipids. It never said anything about a bilayer, so I'm going to say in a bilayer. In fact, I'm almost certain you'll get a mark for saying in a bilayer. If it hasn't specified phospholipid bilayer, 99% sure that that's what it'll want you to say. Ready? Yep, in a bilayer. There we go. Hydrophobic fatty acid tail, point away. Hydrophilic heads, point to water. So I didn't even need to go in this much. I went in too much detail. I would have only needed to say one of those. So I guess I should have started with in a bilayer. Cytoplasm, you could have said. You could have literally gotten 2 marks for drawing a diagram. So I could have drawn that in less detail, put some either side, and gotten it. So let's reword it now. So let's say in a bilayer, the hydrophilic polar head, I won't say glycerol, the hydrophilic polar head faces the water, or points toward would be a better word. Points towards the water. Hydrophobic, non-polar fatty acid chains point inwards away from water. That was a nice question. Ignore You have to ignore... Oh, okay, it says ignore hydrophilic heads, protect hydrophobic tails. That's not the idea. The idea is that they protect each other. That's not the idea. Okay, that makes sense. Let's see how well people did. That was a 2-marker. This question was largely well attempted. Better answers related to the hydrophobic and hydrophilic properties. Question 2 and 3 were highly answered. Let's see. Describe how an ester bond is formed in a phospholipid molecule. Pretty sure this is asking for condensation. Just saying condensation. And then elimination of water molecule to form ester bond between head and chains. That's my answer. Let's see what it says. Condensation between glycerol and fatty acid. Okay, so I might not have gotten all marks there. I said condensation and then I said condensation again. So ester bond between, not head and chain. Silly, not head and chain. Between glycerol and fatty acid. So I might have only gotten a mark there, because it said condensation or loss of water. I thought they would be 2 separate mark points. I explained too much of what condensation was, and I ended up slacking at the end of a question and not putting the right things. So I would have gotten a mark. 2 marks again. State and explain the property of water that helps to prevent temperature increase in a cell. That is going to be high specific heat capacity. And the explanation there is that a large amount of energy is required to raise 1 kilogram of water by 1 degree. High specific heat capacity. Buffers changes in temperature. Okay, so I said that. All I really needed to say was buffers. It says accept ideas such as a lot of energy needed to change temperature. So I gave a physics related answer there. A large amount of energy is required to raise 1 kilogram of water by 1 degrees. So yeah, I genuinely didn't really know that. I think I would have gotten both marks still. But yeah, you literally could have said high specific heat capacity buffers the changes in temperature. Honestly, so far I might have gotten every question right to some degree. Explain how a phagocyte destroys a pathogen present in blood. Come on, we've done this question dozens of times. We know it. So pathogen is engulfed, placed in vesicle. I'm just going to write notes. Vesicle fuses with lysosome to form a phagosome. Releases hydrolytic enzymes via endocytosis. That's the process, endocytosis. Think that's it? Yeah, accept endocytosis. Engulfs, forming vesicle / phagosome and fuses with lysosome. Enzymes digest / hydrolyze. Okay, maybe wouldn't have gotten all marks there, because I said release. I should have also added enzymes. Would it be hydrolyze? Yeah. Enzymes digest / hydrolyze. So maybe not gotten all marks there, because I didn't mention... maybe I would have gotten all marks. Thing I'm noticing, the mark schemes as of recent seem very fluid. This one is not fluid in the slightest. This one seems very... Students should be reminded to use a correct biological terminology. Some students wrote vacuoles rather than vesicles, or confuse the lysosomes with lysozymes. Some students could give a clear sequence of events. People could not read the next question carefully. Give 2 types of cell other than pathogens to stimulate an immune response. This one is going to be foreign cells, or I guess you could say transplants. And I'm going to say cancerous cells. I know what the next question is, I've done it before. Cells from other organisms, yeah, 1 mark. Cells infected by virus, or abnormal / cancer cells. Figure 2 shows the structure of an antibody. Label figure 2 with an X to show where the antigen-antibody complex forms. This one, actually, I got wrong in the exam. You guys know why? Because I do what I always do and I overthink it. What it's wanting you to say is these. That's where it forms. I overthought it and thought, hold on. Why would there be 2 binding sites in an antibody? It must be that. No, it wasn't. I'm an idiot. Don't overthink the question, guys. If you think you got it right, you probably have. A disulfide bridge is labelled in figure 2. What is the role of a disulfide bridge in forming the quaternary structure of an antibody? That's going to be that it holds together each I guess join would be a better word. Joins together each polypeptide chain. That's what I'm going to say. Let's see if it's right. Joins 2 different polypeptides, except holds / attaches. So joins together each polypeptide. Maybe I would have gotten that right. Joins together each polypeptide chain. Joins 2 different polypeptides. I think I would have gotten it right. I guess a better thing would be removing the joins each. Joins polypeptide. Joins polypeptides. I guess maybe I could have said that. So maybe not gotten a mark, but I feel like I should have. Eukaryotic cells produce and release proteins. Outline the role of organelles in the production, transport, and release of proteins. Okay. Don't include details of transcription and translation. Right, let's tread carefully here. This is a question I haven't done before, but I remember similar questions, and I remember what they want you to talk about here. Mitochondria produces ATP for joining of polypeptide bonds. The Golgi modifies protein, releases it, protein, via exocytosis. It's going to be the rough endoplasmic reticulum, contains ribosomes that produce Proteins. Transport and release. Maybe nuclei? Actually, maybe not. Alright, let's check the mark scheme. Oh, really? Oh, yeah. I was going to say DNA in nucleus is code for protein. I was going to put nuclei contains DNA, which codes for protein. Ribosomes / rough endoplasmic reticulum produce protein. Mitochondria produce ATP. I put for joining polypeptide bonds, but actually, I should have been more specific. Thing is, it said don't mention transcription translation, so I stayed clear from it, but it turns out, yeah, for joining, maybe you would have gotten a mark, who knows. Golgi apparatus modify. Vesicles transport. So I said releases protein. Oh, I didn't mention the vesicles. I said releases. Oh man, I rushed it. And then the final one, vesicles fuse with the cell membrane. Okay, so right there, what I did was I took the warning too literally. Yeah, it said don't mention transcription translation, so I stayed too far away from it. I missed out on 2 mark points that I could have gotten if I wasn't so put off by the warning there. So I think I would have gotten 2 or 3 marks out of 4. So still would have done quite well. But, accept exocytosis, so maybe 4 marks. But exocytosis at cell membrane. Okay. So yeah, right there, the nucleus contains DNA, which codes for specific polypeptides. The Golgi modifies and packages the protein. The vesicles transport the proteins and fuse with the cell surface membrane to release them via exocytosis. The area is what actually produces the proteins. Okay, cool. Let's see what they said about this one. Produce good responses. Better answers show that students have read the question carefully, so focused their answers. Okay, the next one apparently caused some difficulty. This is that one question which I got wrong. Suggest why a nucleus is not visible in figure 3. Why would a nucleus not be visible in a micrograph? Maybe the nucleus did not absorb the stain? Will that be an answer? Nucleus not stained. Or, literally the most obvious one, it's a selection. It's a slice, so nucleus in another part of the cell. Okay, this was a question I got wrong. It's vacuole and chloroplast, but I got it wrong because I didn't realise that the blank area was a vacuole. I thought that was cytoplasm. Give one advantage of viewing a biological specimen using a TEM than an SEM. Alright, remember, TEM, the electrons pass through the specimen. SEM, the electrons bounce off the specimen to produce a 3D image. That's a 2D, but as a result, it has higher res. This one has lower res. So one advantage is the higher resolution. TEM has higher resolution in comparison to SEM. That's what I'd say. Higher resolution. Yep, there you go. The cells in figure 4 are part of a continuous layer of cells forming the upper surface of a leaf. The shaded area of the cell is 150 squared. Okay. This is going to be one of those math questions. I hate the math questions in paper 1. They always suck balls. So it's showing you like that. And it's saying this area right here is 150 squared. It's saying the total area of the upper surface of the leaf is 70.65 centimeters. Calculate the number of cells. Alright. The total area. Hold on, this is 2 marks. This is easy. Just convert that to millimeters. So, hold on, if it's you would divide? Yep. So that would be... You would divide by 1,000, so that's 0.15 centimeters squared. And then you would just get the amount of the total volume. 70.65 divided by 0.15 centimeters squared and give it in standard form, which I'm going to predict is going to be about it's going to be about maybe 500. 4.71. Correct answer for any number divided by 1.5. Correct answer was 4.71, which adds up times 10 to the power of 7. Is that really what you would get? 70.65 divided by 0.15. You get 471. Oh! That would be millimeters. So you would have to also divide it by 10 again to get... Hold on, that still would not give you the answer, right? 70.65 divided by 0.015. That's not going to get you. Yeah, that's 4,000. In standard form, that would be 4.71 times 10 to the power of 3. Where have they gotten 10 to the power of 7 from? Accept 1 mark for any answer showing conversion. Did I do it? How did I do it wrong? I'm so confused. The shaded area of the cell is 150 UM. You times it by 10,000. Why would you times it by 100 million? I'm so confused. If you wanted to convert centimeters, hold on. No, maybe maybe I have done something wrong. Hold on. Centimeters, I'd say 70.65 centimeters squared. Let's just assume we're converting backwards. That's going to be 706.5 millimeters. That's also going to be 706.512 divided by 150. That's still not gonna give you the right answer, my guy. Yeah, first 7, that's literally the exact same answer. Yeah, I don't get this. How have I dropped marks on this question? Examiner reports? Proved a challenge, but many students... Bro, this examiner report is not helpful in the slightest. It's literally saying students did well, but some didn't. Like, why? Why did they not do well? Explain. How have I gotten this question wrong? That literally makes zero sense. I'm being completely honest. That's the first exam question I've done in a while, which has genuinely made no sense. How have they gotten the answer? I'm gonna have to go because it's all me to go, but I'll be back later.